Welcome, everyone. It's 12:30, so we will start. We've got, I think several have joined, but at any rate, this is the second WAVEx webinar for this season, and we're pleased to have Dr. Devin Rippner be our guest. He's going to share insights, tips, some various things that he's learned in the last couple years for the 4-acre long-term soil health vineyard that is planted at the WSU IREC in Prosser. And quickly before he starts,
You're muted, Melissa.
there. So I'm Melissa Hanson with the Washington State Wine Commission, and I just want to give a plug for a couple upcoming events in about a month. April 3rd at the WSU Tri-Cities campus, we are combining the WAVE, which is an all-day research seminar, with the WSU Ravenholt lecture series. And we have a guest keynote speaker from Australia, Dr. Mary Ritalik, who's coming. The theme for the day is sustainability meets innovation. And we've got both winery and vineyard topics covered throughout the day. So we hope you will check that out. And then the last WAVEx webinar before the growing season really gets underway is focused on wine tannins. Dr. Jim Harbertson and Dr. Andy Waterhouse from UC Davis teamed together the last 3 years, collaborated on a project, and so these will be some of the results and outcomes of that project. You can register for these events at washingtonwine.org/wave. And with that, thank you. And Devin's going to pull up his screen.
Okay. Can you see my screen, Melissa?
Yep. Good to go.
Perfect.
Thank you.
Well, thank you so much for having me today. It's a real pleasure to be here and to get to speak with folks in the industry. My name is Devin Rippner. I'm a research soil scientist with the USDA-ARS. I'm part of the Horticultural Crops Production and Genetic Improvement Research Unit. Our main location is in Corvallis, Oregon, but I'm at the Prosser worksite. So a little bit about me. So today I'm going to talk about soil health, particularly as it pertains to Washington and Washington vineyards. And I think it's really important to start any discussion around soil health by kind of trying to define it. It is a nebulous term. And so the USDA Natural Resource Conservation Service defines soil health as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. Now, soils are really diverse. There are more than 20,000 soil series in the United States. And this is more types of soil than there are, say, species of vascular plants in North America. So soils can be as different as orchids and cactus compared to, say, a duckweed, right? So totally different plants. We see the same diversity in soils. So in areas with high rainfall, we'll see soils with a lot of iron oxide. So this is a case in the Sierra foothills. This is an alpha sol. In the Yakima Valley area, we see more coarse textured soils, younger soils, no real accumulations of iron oxides. And so here's an example of my daughter a while ago at Hanford Reach, looking at these coarse textured sands. In the Yakima Valley, we see a lot of silt loams and sandy loams that have accumulations of carbonates in them. And then in other areas, there are mollisols. So this is in California, and we do get some mollisols around Prosser. So, soils are really diverse. They have a lot of different properties, and what makes a soil healthy often depends on how we want to use it, right? So what is healthy for, say, blueberries isn't going to be healthy for grapes. And so it's always very important to contextualize soil health around the use of the soil. And so that's why I like to think of soil health as a band that's made up of physical, biological, and chemical components. Sorry, I'm going to add a pointer. Um, and so if you have an orchestra, you can play Metallica. If you have a three-piece band, you can also play Metallica. And so the goal with our soil health band is to get the physical, biological, and chemical components of our soils, our band, to play our soil health music, right? And so that's the goal with management, is to take what we have, what we're given as soil, and try to make it better, more productive, more sustainable for long-term cropping, often, especially with grapes. And so some general properties of a healthy soil are that they anchor life, that they cycle carbon and nutrients, that they're able to provision water to plants. A healthy soil should have limited soil-borne disease. It's hard to imagine a healthy soil that is diseased. And finally, a healthy soil should be resilient to perturbation. And so what that means is when there's a big rain event, you want a soil to be able to quickly soak up all that water. If there's a big heat event, you want the soil to be able to provide good water to your plants. You don't want the soil to blow away in high winds or wash away during a rainstorm. And so a healthy soil is resilient to events, these big storm events, things like that. Now, I think a lot of people, when they think of a healthy soil, often think of like a dark brown, rich soil. Uh, and in some places, sure, this might be what a healthy soil looks like. But our soils often look pretty different than that here in Washington. And these soils are still healthy despite not being necessarily dark brown and looking very rich. So here's an example of a road cut near my house. And so we see this pretty light colored, very mineral soil composed of granite parent material. And we see accumulations of carbonates in the subsoil. And so these accumulations of carbonates, these caliche layers, are just indicative that we live in a really dry, arid climate where water movement tends actually to be up rather than down. And these carbonates are really important in our soil. They hold a lot of our soil together. So here you can see some pedogenic carbonates. So these are just carbonates that have formed in situ that are largely holding this ped together. And so these carbonates actually function in a really powerful role for our soils to make them cohesive. Now, in Washington, kind of what we might want from a vineyard soil is a little bit different than other areas. We're often looking for relatively low soil fertility, often relatively limited water holding capacity, and certainly we want limited soil-borne disease pressure. And the reason for for the low fertility and the limited water holding capacity is we're dependent on irrigation to supply water to our plants. We don't really have rain-fed systems here. And so we need our plants we want to be able to control their vigor and their fruit quality with our inputs, right? And so that's where these kind of lower fertility and more limited water holding capacity uh, properties are important. And certainly, if we have soil-borne disease, that limits the lifetime of our vineyard, which is problematic. So our healthy soil should have limited soil-borne disease pressure. Now, kind of in any context, we can think of some general soil health building practices. And so the first is minimizing disturbance. So here's an example of Dr. Elizabeth Gillespie in Dr. Michelle Moyer's vineyard using a swing arm mower. We're trialing it out. Our soils are dusty. This is part of having a silt loam. But the idea here is that we're able to get rid of the weeds under a vine without really disturbing our soils. So that's a big part of soil health building, is minimizing disturbance. We also want to maximize our soil cover, right? So here are two different vineyards in Washington. One has a pretty established grassy cover crop in the tractor row, and then this one does not. And so when we maximize soil cover, we're able to protect our soils from wind erosion, which is really important. Certainly when you have fruit on the vine and you have a big wind event, minimizing wind erosion is important because the soil particles will actually wear away at the fruit, right? And cause damage to the fruit, which can decrease the value of the fruit. And then finally, the third soil health building practice is maximizing biodiversity. So here's an example of some deer in a vineyard in Lake Chelan. And so, a little bit different type of biodiversity than you might think about. But we can also maximize, say, plant diversity in our vineyard, and then look to maximize insect diversity in our vineyard. So here's a case at Oakville Station in California, where they have some grass cover crops and some mustard cover crops growing. And these attract a variety of different species. And honestly, historically, I've been kind of skeptical of this last one, but I'm going to show some data that kind of highlights the importance of biodiversity in these systems a little bit later in my talk. So with all of this in mind, the state of Washington actually decided to start a soil health initiative. And so they wanted to know and to start doing research and doing extension and making resources available to growers to better understand what makes soils healthy in Washington. And so a big part of this program is this network of long-term research sites that's been set up. And it's the most densely populated network of long-term research sites in the United States. Here's an example of the annual cropping system long-term research site in Western Washington. This is at the NREC. And so there's 8 distinct focus areas. There's a dryland wheat focus area. There's potatoes, tree fruit, irrigated agriculture in the Columbia Basin. Then there's this northwest annual cropping system, LTAR. There's diversified farming in Western Washington. Then we have our wine grape system here. Hopefully in the future, we'll be able to include juice grapes in this. And then there's some focus on the environment and communities. And so right now, these are the 6 research sites. So we have the Mount Vernon site, which I showed a picture of. There's a couple sites, obviously, on the western half of the state. So we have the Puyallup Diversified Organic Systems site. And then we have a number of sites in the eastern part of Washington where a lot of the larger scale agriculture takes place. And so what I'm going to focus on today is talking about the wine grape systems site here at the IREC and Prosser. So this research wouldn't be possible without a lot of contributions from a lot of people. And so, the initial folks who were involved in getting funding for this were Professor Marcus Keller, Professor Michelle Moyer, and Professor Troy Peters. But Dr. Elizabeth Gillespie has been hired in the last 3 years, and she's contributed an enormous amount of time and effort to making this site work. And so I just want to share their names so they're able to get credit for all the hard work that they do. And this is just a small sample of the people that are helping at the site. We are heavily indebted to the folks on our advisory committee, including Dick Boucher and Melissa Hansen, Julie Terrora, and others. And I'll acknowledge all those folks at the end of this. But we're truly grateful for all their support. Here's a picture of our vineyard last year. So it's coming along nicely. So our wine grape systems, LTAR, is on a 4.2 acre plot at the Washington State Irrigated Agriculture Research and Extension Center. Here's a Google Maps photo showing where our plot is. It is located on one soil type, so we're on a Warden silt loam. And we have 5 treatments replicated 4 times on about 0.2 acre plots out there. And our treatments include 3 rows of Chardonnay and 3 rows of Cabernet Sauvignon on 1103 P rootstock. And so we have 168 plants per treatment replicate out at this site, which is pretty cool. Our current treatments are, we have a Washington industry standard out at the site. So this is where we use herbicides under vine and we fertigate for fertility. Then we have some progressively more aggressive soil health building practices implemented at the site. So we have a managed resident vegetation treatment. So this is just where we're not using herbicide under vine, we're just mowing, but we are using fertigation for fertility. We also have an under vine cover crop treatment. And so this is where our cover crop is a subterranean clover legume, and we're mowing this. So we're not using herbicide under vine, but we are then using conventional fertigation. Our next 2 treatments include organic matter additions. So our fourth is organic matter additions. So we are not using herbicide here. We just have resident vegetation under vine. We mow it, and then we're not using fertigation on these plots. We are just using compost or other organic fertilizers for our fertility program. And then finally, we have an integrated treatment. So this includes an alley cover crop and an undervine cover crop. And then we're also not using fertigation out there. So this is kind of our most progressive treatment. Here's a plot map of our vineyard. So we have, working with Department of Ecology, we have 3 wells above and below the water gradient of our vineyard, just to look at how nutrients and carbon and other things are moving into the groundwater. On the border row of our vineyard, we have some rootstock trials. So we have that on the north and south side of our vineyard. We also have border rows on the east and west sides of our treatment plots. And we use these to fill in gaps in our vineyard when we lose vines. We have the ability to control irrigation to the northern and southern half of each treatment plot. And then we have a series of sensors, 3 soil moisture sensors per variety, per treatment replicate. And so we're able to look at the water holding, or we're able to look at the available water in the soil at 3 different depths. So that's 15, 45, and 75 centimeters into the ground. And so, we want to keep track of how water is being used by our vines with these different treatments in place. And hopefully in the future, we'll be able to start including irrigation treatments into our study so we can better understand the effects of irrigation on soil health. We also have a weather station out at the site, and so this really helps us keep track of the weather events that are taking place in our vineyard. And then finally, we have a wind machine on the northern side of our block, and this is designed to help protect all of the vineyards in this part of the station from cold damage during spring cold events. There is a plan over time to build out more vineyards here, so we'll see how that goes. So this is just kind of highlights what we're doing out at the site and our different treatment replicates. So as I said, we're studying soil health at this site. So we're looking at biological, chemical, and physical proxies for soil health. And so we're looking at the phospholipid fatty acids that we find in our soils as a proxy for the microbial communities that are there. We're also looking at mineralizable carbon to better understand the activity of the microbes out there. We're also, in terms of chemical properties, we're looking at available nutrients, as well as total nitrogen, phosphorus, potassium, carbon, and others, just to understand chemical dynamics out there. And then for the physical portion, physical indicators of soil health, we're looking at changes in soil bulk density. So that's the mass per unit volume, aggregate stability, texture, things like that. And the goal of this is to kind of integrate these things so we can better understand, say, like carbon stocks, like how much carbon is there down to 90 centimeters per acre from our different treatment plots. So we can get a better understanding of carbon dynamics in our soil. And eventually, we want to be able to link all of these indicators to plant health and grape yield metrics, as well as juice and wine quality metrics. And so our hope is to better connect kind of the influence of soil health building practices on juice, on grape juice and wine quality, because that's the end goal, right? That's the product that comes out of wine grapes. And so we really want to be able to connect those things. So to get this vineyard started, we did a very comprehensive sampling plan. We collected hundreds of soil cores from the site, both before we planted it and after we planted it. And these soil cores are 36-inch cores that were broken down into 4 depth intervals. So we looked at the top 6 inches of the soil, then from 6 to 12 inches, 12 to 24 inches, and 24 to 36 inches. And so again, we collected these pre-plants. We collected these in about May of 2023. And then we did a fall post-plant sampling where, again, we went out and collected cores. And this time we collected them from the row, and from the tractor row and from the vine row of both the red and white grapevines. in our different treatment plots. And so these were broken down in the same depth intervals. And so this was a lot of work. I'm really grateful for my team for doing this. We also did another comprehensive sampling last fall. That was a lot of work. Kudos to Liz Gillespie for organizing that and getting it done. And so for now, I'm going to show some of the results from our pre and post plant sampling, just to understand, just to show kind of some of the changes that occurred over a pretty short time period, and just to give folks a better idea of what our soils look like at this site, given that they're relatively representative of soils in vineyards in Washington. Here's an example of Brandon Peterson, my former technician, taking soil cores. This was pre-plant. Collins Wakoli, a postdoc in my lab, and Liz Gillespie are out there working on this as well. So it's no joke to take hundreds of these, and we actually end up pulling a lot of soil out of the ground. So kind of to give some global results here. So on this graph right here, along this top x-axis, we have soil carbon percentage, and so it goes from 0 out to 1. And then on this y-axis here, we have depth. So again, as I said, we sampled from 0 to 6 inches, 6 to 12 inches, 12 to 24 inches, and then 24 to 36 inches. The graph basically shows kind of the middle. I set it so that the middle point of each of these depth intervals has the relevant data for that depth interval. And so some of our interesting findings right off the bat is that, one, we have a relatively low amount of total carbon in our soil. So we're well under 1%, looking at around 0.8% total carbon in our soil, and that it decreases with depth, right? So we have less total carbon in our soil as we go down deep. This is pretty normal. The other interesting results are that most of the carbon in our top 6 inches comes from organic carbon sources, right? So this is like plant residues, roots, microbial biomass, things like that. But it decreases rapidly as we go down deep. And what we find is actually after the first 6 inches, most of the soil, most of the carbon in our soil is inorganic. So this is carbon that comes from carbonates, right? And so it really highlights how important this carbonate fraction of carbon is to our total carbon values. And so as we're going forward, we certainly are looking to see how soil carbon changes with depth in our soils, but we're also going to look at how kind of the ratio of organic to inorganic carbon changes, with the goal of protecting our inorganic carbon while increasing our organic carbon. So some other key takeaways that we found here. Here's another graph from data from the site. So this is a graph the microbial diversity and microbial biomass in our soils. So this is just the number of phospholipid fatty acids that we find in our soil right here on the y-axis. And then this is the microbial biomass along the x-axis. I apologize, this is in SI units. It's just how the data came. And it's a little bit funky to try to translate moles of PLFAs to other units in the imperial system. So I just left it as is, because the goal here is for folks to see that each of these different colors. So in pink, we have our 0 to 6 inch interval. In green, we have our 6 to 12 inch interval. In blue, we have our 12 to 24 inch interval. And in purple, we have our 24 to 36 inch interval. And so the key takeaways here are that most of our microbial biomass is in the top 6 inches of the soil. And this is where we find our greatest microbial diversity as well, right? And that as we go deeper in the soil, not only does our microbial diversity decrease, but so does our microbial biomass, where there's functionally not much difference in microbial diversity and biomass below 12 inches in our soil. And so I think this is a pretty interesting thing to see, is that most of the microbes in our soil are in the top 6 inches. That doesn't mean that these microbes that are lower than that are not important, but that certainly our greatest diversity and population of microbes is right near the surface of our soil, which is expected. And we'll be monitoring how these change with our different treatments over time. And I'm grateful to Dr. Garrett Heineck for a lot of help with stats on this. He's been an enormous resource for us as we're processing a large volume of data. Some other interesting results. So these are some results that look at changes over time. So this is here we're looking at our soil organic carbon with depth. Again, so depth is on the y-axis. We have our percent organic carbon on our x-axis here. And one of our interesting findings, and this is actually kind of unusual, is that between pre-plant and post-plant, we actually lost organic carbon in the top 6 inches of our soil. And this is again under vine, so this area was receiving irrigation, and that's probably a large driver of this, right? So microbes were getting water, and they were able to start mineralizing the organic matter that was in this top 6 inches of soil. And so we did lose organic carbon immediately after planting. So within 3 or 4 months of planting, we lost some organic carbon in the top 6 inches of our soil under vine. And we're going to look at how this changes over time, right? To see if this rebounds with time. And certainly, we don't see that loss lower than 6 inches, which is good. Another takeaway is in our alleyway. So pre-plant, this was our bulk density in our vineyard. It was around 1.2 grams per centimeter cubed. This went up closer to 1.4 grams per centimeter cubed in our alleyway. Again, this is totally expected, but this is something that we're going to monitor over time. So this is kind of all the depth intervals averaged together, and we see this increase in our alleyway, in our tractor row, right? And this is not unexpected. We have tractors running through here. They are going to compress the soil. We do see that in this treatment, and we'll be curious to see how our treatments influence bulk density over time. So these are just some quick highlights from our recently established vineyard. And here's 3 photos. This is from 2022, 2023, and 2024. So you can see things are changing very rapidly out at the site, and we're excited to see how those changes continue over the next several years. So with that, after highlighting the work that my group has been doing on the long-term research site, I wanted to also talk about other work that my group has been doing regarding soil health in vineyards in Washington. And so some of this is going to focus on changes at the LTAR at the Prosser IREC, but I'm also going to focus on work on biological disturbance done with Inga Zasada down in Corvallis. So when we talk about soil health in vineyards, we want to minimize physical disturbance, chemical disturbance, and biological disturbance, right? So here's an example of after we deep ripped our vineyard, we pulled out a bunch of PVC pipe, and you can see just hugely disturbed soils, right? Pipe's been ripped out of the ground, big tractor's been putting a lot of force on the ground. So this is an example of massive physical disturbance. I'm not going to focus on this as much, but you can get chemical disturbance in vineyard systems depending on your irrigation, water, and spray programs. And so this is an area that I will focus on more in the future, but I won't really discuss it in my presentation today. And then finally, I'm going to talk about minimizing biological disturbance. Here's a picture from Dr. Michelle Moyer at WSU, just highlighting some phylloxera on a grape root. I'm not going to discuss phylloxera today, but I am going to talk about how soil health relates to northern root knot nematode reproduction success. And so with that, I'm going to kind of break into these other areas. So this topic is going to be on minimizing physical disturbance in your vineyard. This is a very collaborative effort with the folks involved in the LTAR. So that's Marcus, Michelle, and Troy. This work also involves several scientists from the Pacific Northwest National Lab. That's Dr. Tomas Varga and Dr. Anil Batu. And then finally, none of this would be possible without Dr. Elizabeth Gillespie. So I always want to give her a good shout out around this stuff. So when we look at physical disturbance in vineyards, there's some different timelines around disturbance. So there's the establishment phase, right? And so this is often when you get the most disturbance in your vineyard. It could be rotivating for planting a cover crop or something like that prior to vineyard establishment. It could be deep ripping during or just before vineyard establishment. Here you can see PVC pipes getting pulled out of the ground. So a lot of disturbance is happening here. And then there's also continuing disturbance in vineyard. So cultivation under vine is a great method for weed control. And certainly, this contributes to disturbance of our soils. Hilling also happens. This is out at Canoe Ridge. This isn't rootstock, but often if, like in our vineyard, we have rootstock, we hill up in the winter. This is a source of continuing disturbance to our vineyards. And so physical disturbance is just a part and parcel of establishing and maintaining a vineyard. Here's an example of the deep ripping occurring during vineyard establishment. You can see this is quite a large tractor going through here. They're deep ripping the ground. And in the process, they're putting, they're loosening soil right here, but putting quite a bit of pressure on the soil surrounding the shank. And during planting, similarly, our vineyard was mechanically planted by nematodes. We're really grateful for that. And during planting, they put in grapevines, they put in the trellis posts, they put in our irrigation lines, and they put in our trellis wire and wires for our irrigation lines. And so all of this was happening. And during this time, they were opening a large hole in the ground for planting. And this puts quite a bit of pressure on the soil where the tractor tires are. This is a pretty efficient planting process. It's really cool to see. But kind of the end result of this is something that looks like this, right? Where we have this extremely compacted area where the tractor tire was, and this extremely loose area where the vines were being planted. And so just after this, I thought it would be interesting to try to understand what this compaction might look like using X-ray computed tomography, and also to understand kind of what the soils our new vines were experiencing after planting, to see what those looked like using X-ray computed tomography. And I decided to compare them to a field right across the road from our vineyard that contains another vineyard. And so I took several samples from Michelle's research vineyard that is pretty minimally disturbed. It's no-till and mostly hand mowed. Tractors go through it a few times a year, but there's not all that much tractor traffic. And then also from an area next to the vineyard, which was a little bit different than the vineyard soils, but I'm mostly going to focus on the vineyard soils today. So when we look at kind of the effects of vineyard establishment on soil properties, so this would be our minimal disturbance area. This is the area, this is from the field and vineyard across the street from where our vineyard was. We see that soil bulk densities are around 1.4 grams per centimeter cubed in the top 12 inches of the soil. And so this is a decent value. Where we planted our grapevines, or where our grapevines were planted, the bulk density is much lower. This is around 1.2 grams per centimeter cubed. So this is has significantly more porosity than, say, the minimal disturbance area. And then finally, where the tractors were running, we saw significant compaction compared to the other two sites or other two areas. And this is around 1.5 grams, 1.55 grams per centimeter cubed for bulk density. While this is compacted, often in these type of soils, you would think that root growth inhibition starts around 1.6 grams per centimeter cubed. So it was pretty relieving to see that even though these big tractors had been running through our vineyard and had compacted the soil, that the compaction isn't enough to prevent future root growth. So that was really good to see. We also looked at how quickly water would infiltrate in these different soils. So in our minimal disturbance area, we saw kind of a range from, say, like 3 to 8 inches per hour of water flow, which is really good. If we got 3 inches of rain in an hour, it would all flow into the ground. We would have no problem with erosive loss of soils. Where we planted our grapevines, the hydraulic conductivity was much higher. So we were looking on the order of 45 to 80 inches per hour. This is really high. I'm not going to say that it's problematic, but the reality is, like, this water moves very quickly through these soils. it's probably getting down to the bottom of that planting area in minutes. And so it's just something to consider kind of after mechanical planting. Water flows around the vine are going to be very high. And then where the tractor was running during planting, we saw significantly lower water movement, or slower water movement. Sorry, I should say slower. And so we're looking around 0.1 to 1 inch of water per hour. So this would mean we, with even say half an inch of rainfall, something along those lines, we might start having erosive soil loss in these areas. And we're going to keep track of this over time and see how things change with our different treatments. But this was kind of just a baseline understanding of what was happening in our vineyard. Now, I feel really lucky. I was given the opportunity to take some X-rays of the soils, and was able to look at kind of what was the source of these different bulk densities and water flow values. And I'm grateful to Tomas Varga and Anil Batu at PNNL for providing me these images and doing some data analysis. And so on the left here is a soil core taken from kind of the transition zone between annual and perennial grasses in Michelle's vineyard. The middle core is going to be under our vine row, and the far right core is going to be from our tractor row. And so when we look at these, we'll start on the left. So we see lots of roots. That's a root right there. So dark areas here are pores. Some of the lighter gray is organic matter, and then very light gray to white are soil minerals. And so we see lots of pores in this soil. There's evidence. So this is, there were worms moving through this soil. You can actually see the worm castings in the soil column. So let's look at it again. These areas where you see like little BBs in the ground, there's maybe a good example here or up here. So those are worm castings. So these are where worms have moved through this soil. And so this is where the biodiversity component of all this is important. Is, I thought that the low bulk density and high saturated hydraulic conductivity in the soil would probably be due to roots or root channels. And it turns out it's the earthworms that are following the root channels, feeding on them, that contribute to a lot of the porosity in the soil. And to me, that was just fascinating. Now, when we look at this other soil, so this is from under our vine row, we see kind of a mix of porosity in here. So there's not a lot of roots. There are some big pieces of organic matter. I'll show them to you, but we see lots of black areas. So it's just like how the soil minerals and aggregates have kind of resettled has created pores. Here's an example of a piece of organic matter right there, really big. And it's important to discuss that because it influences how we analyze pores in these soils. And then here's where our tractor was running. So we don't see a lot of porosity in this soil. We do see some pieces of organic matter, but in general, it looks like there is fewer pores in this soil. Not completely. It looks like there was a couple root channels right there. But in general, this soil appeared to have lower porosity than the others. So the CT scans are really informative for understanding kind of why these soils are the way they are. Now, I bring up the organic matter because I'm going to show you some data that discusses porosity and pore connectivity in these soils. And the data that I'm going to present was collected using what I would consider normal CT analysis, which does a binary separation. So it just has two components. You either have mineral solids or pores. So this is an example of a soil core that was analyzed using a binary analysis. So you see all the white areas are pores, or at least that's what they would be if you did a binary analysis. But it turns out if you add a third factor, like particulate organic matter, to this analysis, much of the porosity in the soil core is due to these large chunks of organic matter, right? So probably 90% of the pores in this column, what would be considered pores with binary analysis, are actually pieces of organic matter. So here's a piece of organic matter right here. Here's another one. There's a number of these. And so these are just chunks of wood. This part of the vineyard was an orchard previously, and there's just a lot of wood left over. And so we can really see that here. These are just wood pieces. And so my group is doing some basic science here, where we are looking to add this kind of third component to our analysis so that we can do a better job with our pore analysis in these soil columns, because it's really important to distinguish between particulate organic matter and pores. Water is not going to move as quickly through, say, a piece of wood in the soil as it will through a big hole, right? And so that's where it's important. So when we think about the data that we saw in terms of the bulk density and porosity, or bulk density and saturated hydraulic conductivity, so water flow through our soil, we see that the greatest porosity is where the vines were planted, right here in the vine row, and then we have statistically not different porosity in our minimal disturbance in our tractor row soils, and this is largely because of the large pieces of organic matter that are in this tractor row. And so my group is working on trying to sort this out so we can do a better job with the analysis. So even though those two soils had the same porosity, the minimal disturbance and the tractor row soils had the same porosity, they had different pore connectivities, right? And so this is where it's important. This minimal disturbance soil had similar pore connectivity to where our vine row was. So basically we get this middle ground, or we get this scenario where we have the highest water flow, where we have the most connected pores, and then we get the next quickest water flow, where we have similar pores to the tractor row but different pore connectivity. And so this is how these kind of break out in terms of their properties. Is it's based around not only the volume of pores that they have, but how connected those pores are. And these are really insights you can only get from X-ray computed tomography, which I think is really cool. And it was a really neat opportunity to kind of leverage this advanced technology to better understand soil health. I do want to focus on this a little bit more. So worms really alter the properties of soil. Here's an example from my yard. I pulled a pet out. This is about 8 inches long. And what we can see here is that the worms really contribute a lot to the porosity of the soil. You get these extremely long pores that are full of worm castings. Those are the BBs you saw in the CT scan. Here's another example of worms kind of moving through the soil, right? And you can see these areas where there are worm castings all over. And you could see those actually in the X-ray computed tomography scans of the soil. And so I think it's just really fascinating how the worm biodiversity really changes the properties of these soils. So, over time, we're looking for our soil to go from this up here to this down here. And the reality is this is just going to take time. Folks from Cristina Lascano's group started seeing changes in soil chemical properties in 3 years. Let's see, some other folks down in California saw changes to saturated hydraulic conductivity in soils in 5 years. Things may be faster in our vineyard just because we have coarse textured soils, but we're still looking at years for there to be changes in soil health. And so it's important to recognize that changes to soil and soil health take time. If you're curious about any of the papers affiliated with this, or for more information on this, please take a picture of the QR code and you can follow it to different resources. So a big part of this is just finding ways to implement low-till, no-till, and cover cropping practices. Here's an example of strip till. So this is low-till. Sorry, it's loud. This is from Todd Chapman at Coyote Canyon, just doing a strip till along a newly planted vineyard. Here's an example of doing under vine mowing. So this is another no-till practice for weed control. You could adapt like a flail system to take care of that as well. So something that you use for suckering could potentially be used for weed control. And then finally, it's really helpful if you are going to plant cover crops to use a no-till planter. And so there are some links to some Washington-related resources that can help you get access to equipment like this. So kind of, a major question that growers have is how will this affect your grapes? A lot of the research out of California suggests that these low-till, no-till practices with cover crops won't really decrease your yield, but they can increase anthocyanin, so increase grape quality. And there's also potential for decreased pest pressure as they kind of the cover crops can sop up extra nitrogen. And so that can be helpful for dealing with things like insects that feed on grape leaves. So I do realize that I have about 15 minutes left, and I am going a little bit slower than I intended. I'm going to now talk a little bit about minimizing biological disturbance in vineyards and how soil health can play a role in that. Here's an example of some nematode eggs. Postdoc in my lab, working with Dr. Inga Zasada, Dr. Jordan Wade, and Amy Peets down in Corvallis. They did a lot of work studying how soil properties influence nematode parasitism, nematode reproduction using a model system. Here's an example of some nematode eggs. So they had to go through and count these eggs kind of in this field of other debris. It's always really interesting to try to find the nematode eggs. So these are them right there. So with that, I'm going to go into this a little bit. So we had this hypothesis that healthy soils can reduce pest pressure. And our thought was, if soil properties didn't have an influence on pest pressure, then when we inoculated our experimental crop, so we use tomatoes for this work, just because that's the model system that Inga has going. So we grew tomatoes in soils that we sampled from all over Washington State and one soil from Oregon. And the idea here is that if soil properties don't influence nematode reproduction, then what we would see is all these circles would be the same size on this map. But we see that soil properties actually have a huge influence on nematode reproduction success. And so we found this really fascinating, that there were like these inherent soil properties that seem to control nematode reproduction. And so we were curious about how we could kind of tease apart our soil test data and our nematode egg count data and try to find which properties were most influential in controlling nematode reproduction success. And so a big part of this was trying to see if there were soil health test indicators that were relevant to this. And so if you get a soil health test, you might see some of these terms. So you might see active carbon. This is permanganate oxidizable carbon. You might see something like aggregate stability, ACE protein. So this is autoclavable citrate extractable protein. You might get like a soil respiration. So this is mineralized carbon. It's respiration from the soil. You might, get a test for soil organic carbon and nitrogen, or a test to understand how much nitrogen is mineralized, made plant available over a period of time. These are all pretty well established soil health test indicators. Some of them are more chemical, some of them are more biological, some of them are physical, but all of these are widely considered to be important indicators of soil health. And so our analysis really focused on these indicators to see how important they were for nematode control. I think for this, I'm mostly going to focus on active carbon, POC and soil respiration. So this is mineralizable carbon. These were found to play a pretty big role in inhibiting nematode reproduction success. One of the things I am going to go talk a little bit about these is soil health indicators. So active carbon, POC, it's a bit of a misnomer. So POC is actually likely an indicator of plant carbon. There have been some really interesting studies that have come out in the last few years where they basically dosed quartz with sugars and lignin and tannins and all these other things, and then reacted them with permanganate oxidizable carbon just to see what it was measuring. As it turns out, it's really sensitive to measuring things like lignin and organic acids like tannic acids, but it doesn't actually react with sugar. So glucose had a similar reaction to just quartz. And so this suggests that permanganate oxidizable carbon is not actually like an active carbon indicator. It's an indicator of like microbially processed carbon or carbon that is harder for microbes to process. And this makes sense. So like the origin of this method actually comes out from studying dairy feed. And so it was used to indicate how digestible feed was for cows, and feed with high POC was not very digestible. And if you think about cows as being this like giant bioreactor, then it makes sense that Epoxy is maybe not active carbon, but more of this like recalcitrant stable carbon. Now, grape pumice could be a great source of the aromatic compounds that are often highly reactive with epoxy, and I would love to do some studies in the future where we look at how grape pumice might affect root knot nematode reproduction success. I think that'd be a fun way to take this study, or some of the studies that we've done. So maybe a better indicator of active carbon is this, like in your soil, is this mineralizable carbon. So this is a measure of soil respiration, CO2 generation from the soil. So basically what's done is soils are incubated with water at a certain temperature, and then they just measure how much CO2 evolves. And so when microbes with water come in contact with sugars or other things that they can break down, they're going to produce CO2. And so this is seemingly a better indicator of active carbon in soil than epoxy. So I just wanted to kind of put that out there as kind of an educational moment around these different soil health metrics. So basically what we found, like the key takeaway from this study, is that there are some soil health indicators such as epoxy or mineralizable carbon that seemingly play a big role in decreasing nematode reproduction success. Let's see if I can get this. So what we find is that some of these organic matter cycling indicators, that's how we call what we call them, using the statistical method called what is it? SEM, structural equation modeling. So we do find that these organic matter cycling indicators seemingly have a negative effect, a significantly negative effect on nematode reproduction success. Some of our indicators for like soil structure and moisture retention are not significantly involved in this, but they, the model did spit out that they were relevant, even though it was not significant. So we find this interesting. Like, I think it's really cool that there's an association between these soil health indicators and plant parasitism success. And so it's fun to kind of look at how these indicators spread out in Washington and Oregon. So one of our key takeaways is it seems like increasing permanganate oxidizable carbon was associated with decreased nematode parasitism success in Washington and Oregon. When we really look closely in the Yakima Valley and some of the bigger wine growing regions of Washington, Columbia Gorge, Walla Walla, we tend to see that areas with higher permanganate oxidizable carbon have lower nematode reproduction success, even when things are in pretty close physical proximity to each other. So soils that just have higher permanganate oxidizable carbon seem to inhibit nematode reproduction success. Similarly, mineralizable carbon seems to play a role. So we do see often, when there's more mineralizable carbon, we had less success. The size of these circles is correlated or directly relates to the number of nematode eggs that we counted per gram root during our experiment in these soils. And it also seems like pH plays a role. And it's cool to see this. Catherine, Inga, Jungmin, and Paul Schreiner and I did a study previously where we did see, especially with Washington soils, that soil pH did seem to play a role in northern root knot nematode reproduction success. And so it's cool that we saw similar results in our larger experiment where we sample from all across the state. It's always nice when theoretical studies line up with more practical studies. And if you're curious about the results from this work, please follow the QR code here. So the key takeaways here are that increasing your soil health can decrease nematode reproduction success. So keep working on building soil health. It can improve a variety of things in your vineyard, including making it potentially making it less vulnerable to nematode plant parasitism. Just keep minimizing disturbance and enhancing soil health. And with that, I'm done. I am very grateful to the folks here. I can't thank them enough. We have our advisory committee, Julie Terrara, Sarah Del Moro, Cody Hartley, Patrick Ron, Dick Boucher, Matthias Kuslis, Catherine Jones, Melissa Hansen. None of this stuff would be possible without them. Liz Gillespie. I mean, I could just go through this list. But yeah, I'm just grateful for everyone that has contributed to my success here in Washington, including the folks at the Pacific Northwest National Lab, the Washington Wine Commission. I am indebted to their funding. I'm so grateful that they have been willing to fund our vineyard and help us get it off the ground. And here's a picture from last year of my lab group with my son. So anyway, I'm happy to take any questions.
So if you want to put your questions in the Q&A, I will read them to Devin. And if not, I realize we're right at the time. Everybody who registered will get the link first. Then we will be putting the recording up on the Wine Commission's website. And so when you get the link, if there's a burning question that you thought of, send it to me or reach out to Devin directly. You can find him on the internet. So with that, I do not see any questions. So we will close this down. Thank you all, and thank you, Devin. And I hope to see you at the WAVE Ravenholt Sustainable Seminar or the next WAVEx in May. So thanks again.
Thanks, everyone.